in the 19th century. In the middle, of course, I have the many images of the coronavirus, which you will now find on the internet. Now let's take a look at the coronavirus pandemic as of July the 1st, 2021. The burden of the coronavirus pandemic is enormous. About a month ago, more than 4 million people had already died, and more are dying even today. India and the United States together account for about 1 million deaths between the two countries. And in the figures that I show you here, which are taken from the Johns Hopkins dashboard, you will see that the daily cases since the beginning of 2020, which are those red graphs, show you the waves of the pandemic. In India, for example, the pandemic has already gone through 2 phases. The second one was a somewhat narrower phase, but a much more intense phase. The first phase was a somewhat broader phase. And this is because the virus has mutated between the first phase of the pandemic and the second phase of the pandemic. We are still in the midst of the pandemic. China, of course, has reported very few cases so far, but today it appears to be increasing. Now you might ask, why should one actually study the coronavirus? And what should we learn about the coronavirus? I've taken a slide which will give you the reason. And this is taken...
So, Balram, slides are not changing.
Slides are not changing. Okay. Let me see. Just a moment. Are they changing now?
Now we can't see anything, sir.
Can
you see now?
Yes,
Yes. Okay. What you're not getting on the slide, which
Is sharing?
are you seeing a blank slide?
Nothing, sir. We are not seeing anything. No slides.
Yeah, there's no slides, sir. Yeah.
Yeah. Is it black?
Nothing, sir. No.
Nothing appears, sir.
Nothing appears. Okay. Then I'll move from that slide because it should appear. Can you see the slide now?
No.
No, sir, I think sharing has to be done again.
Again?
Yeah. And then that too, that slide, a presentation mode.
I'll go back and escape again.
Yeah,
Yes,
please.
And come back and share what's... Let me just see, how do I do that? Go back here and present window.
Hello.
Has it begun to share now?
It's starting now.
Yes, sir. Yes.
Okay. I'll make it full screen.
Yes, sir.
You see? Is it moving?
No, sir, we can see the first slide, Rohi, and that too is still not in the presentation mode.
It's on my screen, it's on the presentation mode. Has it moved?
No, sir, not yet.
So there's some problem, probably. Connection seems to be fine. Because you can see me and I can see you.
Yes, sir. We can see the slides also, but they're not moving.
Okay, sir, can I speak?
Okay.
Sir, Balram sir, first you go to the presentation mode before sharing the slides, sir.
Yeah, I've gone to the presentation mode.
No, first you stop sharing.
Okay, and
Stop sharing.
okay.
And then go on your screen presentation mode.
Now I'm sharing. Can you see it now?
Yeah.
Let me see if it moves now.
No, sir, no.
In this mode, it doesn't move. I'll make it full screen. Let me see.
Yes.
Yeah. Can you see it now?
No,
Still
full screen is not coming.
stuck up first slide only, sir.
Full screen भी नहीं है.
Full. So before you share, please like convert it into. You just press that presentation mode.
Other mode is we can share the entire screen. That also works sometimes.
Yes, sir.
Let me try that way.
Instead of sharing a
Usually doesn't work that way. Let me see. Maybe Google Meet works that way.
Entire screen will be focused on window.
हम अखबार का मेल करवा दें.
Or else you can send an email, sir. Kindly say, if you don't mind, send your slides. We'll present it here.
Okay, let's try one last time. Can you see?
Yeah, please, sir. Yeah, do it once. Yeah.
It doesn't move in that mode.
Yeah, actually, it's not moving. That's the trouble. Yeah.
Yeah, now in this mode it's moving, but you are not. It doesn't move at your side.
Yes, sir.
Yeah, here it moves. All right. I'm surprised. Okay, let me just try. You suggested that I do something else. No? What was that? Would it help if I went to full screen first?
Yes, sir.
go
And
to full screen,
within
then
that full
share that particular window.
yes, sir.
I've been going to one window. This is now my entire screen. You'll see yourselves. It doesn't work. You get only the first slide and then it doesn't move. That's the problem, I think.
Yes, sir.
Do you see the first slide on the screen?
Yes.
We are able to see, but it's still not in the presentation mode, sir. The
Okay,
first
now,
slide.
now I've gone to presentation mode.
Okay, sir.
Do you see it on the presentation mode now?
No, sir. I think there's some issue.
Yeah, there's some problem at this point.
Yes, sir. Sir, could you please, you close this window first, the one you are sharing.
Okay.
Okay, then you keep your slides in presentation mode on your desktop open.
Okay, let me do
Minimize
that
that window.
one. Yeah. We see if that works. Yeah, I'll go to full screen.
Yes.
Then escape from this.
You have to escape. You have to stop presenting. First, you have to stop presenting the previous one.
Yes, sir.
That is not done. First, he has to stop the presentation. Then only you have to restart the presentation.
Yes, sir. यूट्यूब का लिंक दे दो, यूट्यूब का लिंक शेयर करिए आपको भी। अब तो सर खुद भी नहीं दिख रहा है, यादविंदर। Some.
Second.
अभी म्यूट ये से
अभी नहीं सर, आर यू देयर? अभी है क्या वो? म्यूट रखा है। अभी मत करो, डिनाई करो, नीचे मैसेज कर दो, यूट्यूब
लोगों को कर दीजिए।
हाँ जी, कर लिया उनको।
बाहर वालों को मत करिए, अपने यहाँ के जो है उन्हीं को डिस्कनेक्ट। हाँ है।
Now he is present. Again the same issue.
If you go to the second side, no, then it will be fine. If you go to another side, even like this also it is fine.
So please move to the next slide.
And I've turned off my mic.
Okay.
This is confusion. Yeah. I'll go to full screen now.
Okay, sir.
I'll go to full screen. Now I think it will work because I was seeing many things on my screen before.
Okay, sir. Yeah. बहुत सारी चीजें हमको अपनी स्क्रीन की दिख रही हैं।
Can you see the slide now?
Yeah, slide is visible, but not in full screen. Anyway, if you can.
Anyway, sir, please keep on moving to the next slide so that we can manage this way.
On my slide, on my screen, it's full screen. Okay, so what I'm going to tell you is a little bit of the history of the coronavirus, little chemistry, some biology, and then towards the end, I'll talk a little bit about science and evolution. I've pictured Darwin on the left, on the right, and Mendelian on the left, just to illustrate that these are the foundational pillars of chemistry and biology. Can you see the second slide now?
Sir, again some issue, sir. I don't know what is happening.
Again some issue.
Yes, sir. Even if you go to the next slide, it's not
Face down, face down if you can do it. And
Yes.
the slides will move.
Yes.
Sorry to interrupt you, sir. Please use your cursor at the
I'm
second
focused.
slide here, so definitely it will work.
This is my second slide now. Can you see it, or you're still seeing the first
No,
one?
sir. Please come out from this full screen mode, sir, because you are in the full screen mode, but that thing is not here.
Yeah. Now I'm on full screen mode here.
Please come out from the full screen and just use, click the cursor on the second slide. Yes, now it works.
Yeah, now it's coming. Now it-
Yes.
This way we can do, sir. It's comfortable.
Okay. I'll work without the full screen mode then. Okay. Can you see the second one now?
Yeah,
Yeah.
sir, of course.
Okay, so the second slide is the slide which just tells you what the burden of the coronavirus pandemic is. And most of you are familiar with this because you would have seen it repeatedly in the newspapers. Now I'll skip my third slide and go to the fourth slide. Since the pandemic started last March in India, people have really been intrigued by the coronavirus. It captured everybody's imagination at the beginning. And all these pictures used to appear in the newspapers where you had policemen, auto rickshaw drivers. In Europe, you had chefs, all of them making helmets and images which resembled the coronavirus. So what I'm going to tell you is a little bit about how the coronaviruses were discovered and some things about the present coronavirus. Last year in March, when the first lockdown was announced, it was a new experience for most of us. So when we were completely locked inside the house for days together, I began to read about the coronavirus. And I asked myself a question: who discovered the coronavirus? And at the end of my search on the internet, I wrote a couple of articles, both of which appeared in the magazine Frontline, which is not a science magazine, but a popular magazine. But a little while later, the articles that I wrote appeared in a newsletter in Ouray, Colorado, which is a small town with a population of 1,000 people up in the Colorado mountains. So this part of my talk could be entitled How I Spent Time in the Total Lockdown, or How I Came to Feature in a Newsletter in Ouray, Colorado. What I did here was to simply trace the old history of the coronavirus. After I had done this, I was interviewed by an archivist in a library in the Northern Arizona University. And at that time, I told her this. I said that it is a tribute to the power of the internet that the road from Ouray to Flagstaff, both of these in America, passed through Bangalore. It also seems to me that in some strange way, the virus has led us to its discoverer by turning the world upside down. Because if we had not been locked up in a room, we would not be searching the internet looking for who discovered the coronavirus. But first, let's define a virus. One of the best definitions of a virus that I've seen is in a philosophical dictionary of biology written by the English immunologist Peter Medawar. He calls his dictionary Aristotle to zoos. And here he defines a virus. He says that inasmuch as viruses are made known only by their causing disease or other pathological changes, the existence of benign viruses having no ill effects remains conjectural. No virus is known to do any good. It has been well said that a virus is a piece of bad news wrapped up in protein. What is wrapped up in protein? It's the nucleic acid. In the case of the coronavirus, ribonucleic acid or RNA, which is the genetic material, the instructions for replication. This is now wrapped in a protein coat and a phospholipid bilayer. The phospholipid bilayer is important because it is in this bilayer that the coronaviral spike is embedded. So the coronavirus is an enveloped virus. It has a bilayer phospholipid membrane. It picks up the phospholipid from the host cell. And the virus protein, which we call the spike protein, projects out from the surface. That gives it the characteristic image. There is a corona around the spherical object. At the beginning of the pandemic, we were told that we must wash our hands very frequently and use sanitizers, which mostly contain alcohols. The reason for this, of course, is, as William Osler said more than a century ago, he's one of the founding fathers of modern medicine. He said that soap and water and common sense are the best disinfectants. And that is true even today. So let's ask a question. How was the coronavirus discovered? The first picture of the coronavirus under an electron microscope emerged in this paper in 1967. This paper had a title, The Morphology of 3 Previously Uncharacterized Human Respiratory Viruses, that grow in organ culture. The authors were David Terrell and June Almeida. Last year, National Geographic and then Scientific American and later all the newspapers carried articles on June Almeida as the woman who discovered the coronavirus. What she had done was to take the pictures which are shown in the center of this slide. So I read this paper, and what I have marked and read is the most important thing, because from these experiments, what came out was two human viruses called 229E and B814 are morphologically identical with avian infectious bronchitis virus. So here was another new respiratory virus, which had those characteristic spikes on its surface. 229E is a number that you should keep in your mind, because when you read a paper, it's useful to read the paper in detail, useful to read all the figure legends and even the acknowledgements. If you look at the figure legend, it says that the particle which was imaged here is strain 229E, and the present technique was electron microscopy, which was in its infancy in 1967. And then if you read the acknowledgements, it says, "We wish to thank so-and-so," and then it says it thanks Dr. D. Hamre for the 229E virus. So when I read this paper, I realized that the virus had actually been isolated by Dr. D. Hamre. And now who is Dr. D. Hamre? So I went back to the literature and I found this paper in 1966, 1 year earlier, where a new virus had been isolated from the human respiratory tract. And the full name was here, Dorothy Hamre. So I realized it was a lady. And the virus had actually been isolated by a lady as far back as 1966. What she was doing was she was looking at nasal secretions from medical students who would come to the dispensary at the University of Chicago. And she was collecting the nasal secretions and trying to isolate new viruses. Today it is known that 30% of common colds are caused by coronaviruses. So the coronaviruses were original viruses were isolated from people suffering from a common cold. But Dorothy Hamre's paper was remarkable. She treated the virus with 5-fluorodeoxyuridine and showed that these inhibit the virus. This meant that the nucleic acid in these viruses is RNA. And using classical techniques of filtration, which were available in the 60s, she determined the size of the virus as 89 millimicrons, what we will today say 89 nanometers. Occasionally, this virus has been shown to cause acute respiratory distress, just like the current coronavirus. And there's a 2018 paper to show this. If you now move to the 1967 paper, you find that they have also measured the average diameter from electron microscopy, and it comes out to between 80 to 100 nanometers. Remarkable coincidence between Dorothy Hamre's measurement of the size and David Terrell's measurement of the size. So by 1968, scientists had written a letter to the editor of the journal Nature, where they said that there is a new group of viruses called the coronaviruses. And if you look at the names of the people who wrote the letter, you will find the name of D. Hamre. But some years later, 7 years later, when the formal name came into the literature, you will find that her name has now disappeared. So I was puzzled here. I asked myself the question, who was D. Hamre and why is it that nobody knows anything about her? So I went back to Google, to PubMed, and to Google Scholar and searched the literature. Remember, last year from the middle of March till the middle of May, we were locked in. So one had to stay at home. Now, when I looked through Google, I found many papers written by Dorothy Hamre, all of these in the period between the late 1930s and the 1970s. And on PubMed, I found that she'd published 40 papers between 1943 and 1972. And if you look at all these papers, you find that you have a profile of a productive scientist beginning in bacteriology in 1941, transitioning to virology, and having a publishing career which spanned 30 years. I was interested in finding her picture, but I couldn't find a single picture on Google Images. So I asked the question, who was Dorothy Hamre? There were other people called Dorothy Hamre. None of them was old enough. None of them was a virologist. And therefore, I began to search further. And suddenly I found her name in a collection in an archives in the US state of Arizona at Northern Arizona University. And the creator of this archives was a person called Alexander Brownlee. But Dorothy Hamre's name was also there. And then I realized that Brownlee was her husband. So I wrote to the curator saying that I'm interested in finding a photograph of this person. And I told them that she was probably the discoverer of the coronavirus. So they took a lot of interest, and I won't go into this long story, but eventually we were able to find pictures of her, and I now show you a picture. This was taken at the University of Chicago in the 1960s. She retired rather early and went away and settled in Ouray, Colorado, where she died. And that is why my article now appeared in a newspaper in the small town in which she died. But I bring this to your attention to tell you that sometimes there are stories in the scientific literature which are worth looking at. Here was a lady. There were very few ladies getting their PhDs in America in the 1930s. Not only did she get a PhD, but she had a very productive career. She was an expert on the rhinoviruses. She discovered the coronavirus, yet she retired in the early 1970s with the title of research associate. So somebody who's begun a career during the Depression, worked through the Second World War, all the way to the early 1970s, never ever got a position on the faculty. This, of course, was the case for women in many parts of the West and many parts of the world. So we now know how the coronavirus was discovered from nasal secretions. But why were people looking at nasal secretions? David Terrell wrote a book in 2002 called The Cold Wars, and this was the fight against the common cold. Because after the Second World War, most loss of labor at industrial factories was because people were suffering from colds and they wouldn't come to work. So everybody was interested in a cold vaccine. Today we are interested in a coronavirus vaccine. A cold vaccine would be a rhinovirus vaccine, and also possibly a coronavirus vaccine. And what they did was they took these nasal secretions from people and put them into healthy people to see whether they got cold. So they would get volunteers by advertising. They would put up advertisements like this saying that if you come here, we'll see if you catch a cold, but even if you do, we'll look after you. Otherwise, you will spend 10 days on a free holiday here. And this site is now no longer in existence, but this is the way research was done. And this was remarkable research from which a great deal of work about respiratory viruses rests on work like this. In 1996, David Terrell wrote a review, a chapter on coronaviruses in a textbook of medical microbiology. And he concluded that coronaviruses cause acute, mild upper respiratory infection, common cold. And with that one statement, most people lost interest in research on coronaviruses because they didn't seem to be causing you any great problem. One digression here. You can ask a question, are viruses living or are they just chemicals? You will find arguments on both sides in the literature. I put up a picture here which says 10 reasons to exclude viruses from the tree of life. And below I put another paper which says, ask the question, do viruses exchange genes across the super kingdoms of life? So in a way, viruses are mediators of gene transfer sometimes. But in 2003, the first SARS outbreak took place. This is what we today call SARS-1 or SARS-CoV-1. Today's virus is SARS-CoV-2. There was an outbreak of respiratory illness in southern China. And then there was a call to the World Health Organization on the 28th of February in 2003 from the Vietnam French Hospital of Hanoi. They said, we have a patient who appears to be sinking. He has a new kind of infection. WHO immediately sent a doctor. This doctor is one of the most remarkable people in modern medicine, Carlo Urbani. Young man then, he went across immediately to Hanoi and within a few days recognized that here was a new infectious disease. He immediately got the government of Vietnam to impose extraordinary quarantine measures. And this is how the first SARS outbreak was actually contained. But in that time it had spread. Urbani was infected. He flew to Bangkok. But there is the story of him sitting in Bangkok airport, not allowing anybody to come near him, asking for a PPE at that time, putting it on, and he died a few days later in a Bangkok hospital. All put together, there were 8,000 infections and about 800 deaths. So it was a very severe virus. 10% of the people infected died. The current virus has not killed so many people percentage-wise, but it has infected more people. So at this point, I will have a short digression into chemistry to tell you why chemistry is important. Chemistry is the subject that I studied as a young man, and it's a subject that I still like very much. The best definition of chemistry is in this article. Arthur Kornberg, the Nobel Prize-winning biochemist who discovered the enzyme DNA polymerase, he fired, I would believe, the first shot of the biotechnology revolution. He described chemistry as the lingua franca of the medical and biological sciences. One of the saddest things of medical education today in India is that very little chemistry is studied there. And even more sadly, even among the education of biological scientists, chemistry does not occupy a very important position. Chemistry deals with matter, and matter is made up of atoms. And since some of you might have been trained in physics, I take you back to the very first of the Feynman lectures. First lecture, matter is made up of atoms. And Feynman asks a very interesting question here. If in some cataclysm all of scientific knowledge were to be destroyed and only one sentence passed on to the next generation of creatures, what statement would contain the most information in the fewest words? He answers this question, and that is in red. He says, I believe it is the atomic hypothesis or the atomic fact, or whatever you wish to call it, that all things are made up of atoms, little particles that move around in perpetual motion, attracting each other when they are a little distance apart, but repelling upon being squeezed into one another. This is, of course, the principle of the force between two atoms, neutral atoms as they move to form a molecule, the basis of bonding, and also the basis of van der Waals interactions in chemistry and also in biology. Now, on a chemical scale, biological objects are just large chemicals. So what I have here are the length and time scales that one encounters in chemistry and biology, and sometimes also in physics when you apply physics to problems of chemistry and biology. Look at the length scales and look at the time scales. The virus, for example, which is right in the center of this picture, lies somewhere between the ribosome and mitochondria. So if you're studying biology, you will immediately be familiar with the protein synthesizing machinery in cells, which is the ribosome. The structure determination is what fetched Venki Ramakrishnan the Nobel Prize, and mitochondria, where energy is generated in the cell. The importance of chemistry is illustrated in this cartoon. This gentleman is doing a crossword puzzle, and he asks the lady, "What's a 9-letter word for biotechnology?" She answers, "Chemistry." So I will also ask you a question. What is a 9-letter word for material science? This will also be chemistry. And then you will find that there is nothing around you, nothing in the world, including yourselves, which is not made of chemicals. So you have nothing which is chemical free. So if Patanjali promises you something which is chemical free, or Tata Salt promises you something that is chemical free, they don't know what chemistry is. But chemistry and mathematics are two subjects which children find difficult in school and even later. Only some people go to chemistry and some people go to mathematics. And physics and biology are easier to relate to simply because we understand motion. We have an instinctive feeling for gravity. Biology, we see animals, plants all around us, and therefore we appreciate diversity in biology. But in order to study chemistry or mathematics, you need to learn symbols. You need to learn a language, and you need to learn new alphabets. Chemical structures are the alphabets of chemistry. Symbols and equations are the alphabets of mathematics. But amazingly, in India, politicians understand both chemistry and arithmetic very well. The Prime Minister, for example, said after the last Lok Sabha election that chemistry defeated arithmetic in the 2019 polls. And he was right. And you will find many other politicians have also used the metaphor of chemistry to explain when they win something, it's good chemistry. If they lose, it is bad chemistry. So you know what chemistry is intuitively. The foundational pillars of chemistry. Diversity, illustrated by Mendeleev and his periodic table. Unity, illustrated by Wohler and his famous synthesis of urea from ammonium cyanate. If you remember, those of you who have studied a little bit of history in chemistry will realize that he immediately wrote an excited mail to another famous chemist, a letter in those days, to Berzelius, in which he says that I have now made urea without the help of kidneys, man or dog. Because urea was considered a biological molecule, whereas ammonium cyanate was considered an inorganic molecule. And there was this big gulf between inorganic and organic chemistry. Wohler symbolizes unity. Boltzmann, and I'll come back to him, symbolizes dynamics, because Boltzmann is the man who first had the idea of the atom. He also gave us the idea of entropy. And then, of course, Pasteur, who we remember more in microbiology. But Pasteur is also remembered in organic chemistry because he gave us the earliest ideas of stereochemistry with its famous resolution of tartaric acid. So chemistry now is very diverse, lots of elements, lots of molecules. You might ask, where do all these elements come from? They come from the stars. They came on Earth when it was formed. This is why evolution is so important. How did life evolve on Earth? From a beginning in which there was no life. I draw your attention to this book, which is Jacob Ronowski's Ascent of Man. This is a book which is based on a television series which appeared in the 1970s. but it's a marvelous book in which Ronowski, a theoretical physicist, traces the ascent of man. And this is the ascent of man right from the beginning, from the time man became civilized or began to get civilized, all the way till today. And I'll come back to this right at the very end. But he says here that in all the stars there are going on processes which build up the atoms one by one into more complex structures. And then the sentence in red, matter itself evolves. Then he says, the word comes from Darwinian biology, but it is the word that changed physics in my lifetime. Every one of you who is listening must get your own meaning for the English word evolve. What does evolution mean? He talks in poetic fashion about carbon, for instance, because without carbon there is no organic chemistry. Without organic molecules there is no life. He says carbon, for instance, is formed in a star when 3 helium nuclei collide at one spot within less than 1/1,000,000 of a millionth of a second, 10 to the minus 12 of a second. Then he adds, every carbon atom in every living creature has been formed by such a wildly improbable collision. So you realize when you read this that life itself is highly improbable. And if we ask questions about the origin of life, we are asking questions about the origin of chemistry, and then the origin of biochemistry, and eventually the origin of biology. We will return to the virus. After Urbani discovered a new infectious disease, people began to work on it. And now once again, you will see electron micrographs of the coronavirus. And by this time, electron cryomicroscopy was also available in the year 2006. We've come to the 21st century. And the particle diameters were now estimated. And you can see with this new technique, 82 to 94 nanometers. If you remember Dorothy Hamray's 1966 estimate, 89 nanometers. So you can see she was spot on, right in the middle. So we can only now pay a tribute to the people who worked in the 1960s identifying the coronaviruses. Today with techniques of microscopy, one is able to get a much better picture of the arrangements of the protein and nucleic acid components of the coronavirus. You can measure distances between them and get a detailed picture of the virus itself. These pictures were available. They were available before this pandemic, except that nobody was interested in developing a vaccine because there was no disease. SARS-CoV-1 had disappeared. Now it's only after SARS-CoV-2 came has there been this mad rush now to make vaccines. This illustrates the importance of keeping research going without always asking the question whether that research is useful or not. This would have been extraordinarily valuable research to take towards a vaccine. Now after 4 million deaths and probably 5 million deaths, and if the figures are underestimates, maybe 10 million deaths, maybe governments and those who support science will learn a lesson. Now, in the process of viral fusion, the most important protein is the spike protein. For developing vaccines, the most important protein is the spike protein of the coronavirus, what projects out from the surface. Proteins are important, but sometimes we forget that. So I have this little slide here. I quote the high priest of molecular biology as early as 1958. He says the main function of proteins is to act as enzymes. And then he says it is at first sight paradoxical that it is probably easier for an organism to produce a new protein than to produce a new small molecule. That is, metabolites are more difficult to make than small molecules. And then he says there seems little point in genes doing anything else but protein synthesis. This is very important. The main function of genes is really to produce proteins. The most important step in infection is the virus cell fusion step. Much of our understanding of the fusion of a viral membrane with a host cell membrane is derived from early work done on influenza virus. Remarkable work done over the last 20-25 years in which The structure of the influenza virus hemagglutinin, which projects from the surface, has been done, and its role in membrane fusion established. And models like this are now available in the literature. It's these models that we now extrapolate to the coronavirus. On the next slide, I show you the pictures of the current coronavirus spike protein. They've all been determined. There's been a remarkable outpouring of research from January of 2020, continuing today. It's been a deluge of papers on the coronavirus. So detailed atomic level structures are now available. So we can understand a great deal about the mechanisms of viral recognition and viral infection. If you look in detail, the coronavirus spike protein is a trimer. So the picture on the right is of the trimeric protein. The picture on the left, I've taken out one of those and shown it to you. It's a complex protein, multiple domains, and you will find these terms even in the newspapers nowadays. RBD, receptor binding domain. That's the part which recognizes the human cell, the angiotensin converting enzyme, which is the receptor for the coronavirus. So we know the structure of this protein in detail. And now not only is the structure of this protein available in detail, but the structure of the binding domain with the receptor protein is also available in detail. Again, determined last year. So this is all very recent science. Now, when you have these detailed pictures, how is it going to help you? And I'll show you that in a couple of slides. Because once you have such enormously good structures, you can ask, how can we interfere with this process? That would be, how do we make therapeutics or vaccines? How can we catch the spike protein before it binds? Or how can we prevent the spike protein from binding? These are the questions that we actually ask. So this is the kind of science which needs to be done in order for people to come to rational ways of finding treatments for other viruses in the future. Now look at the daily cases now again to show you. I compare two countries, India and the United Kingdom. Now, of course, in the United Kingdom, the Indian cricket team is there. Everybody is playing. Wimbledon was over. The Euro soccer tournament was over. So people are mixing. And you can see that towards the right side of the bottom part of the graph, it is slowly climbing up. It will climb up, although people have now been vaccinated. In India too, it has come down, but it is slowly beginning to go up again. I'll go back now and digress to biology. You might ask, what do we study in science? What we study in science is really we study nature. We study natural phenomena. Physicists study natural phenomena, biologists study natural phenomena, even chemists study natural phenomena because they deal with the materials which are responsible for the phenomena. There are two very prestigious journals in science. They're called Nature and Science. The journal Nature, and you go back all the way to volume 1, number 1, the year 1869, when Nature first started. They published in their first issue an editorial. They asked the famous biologist Thomas Huxley to write this editorial. But what Huxley did was he didn't write the editorial. Instead, he translated from the German an essay which had been written by the German poet von Goethe. And I just quote one sentence from Goethe. He defines what is meant by nature. He says, we are surrounded and embraced by her, powerless to separate ourselves from her, and powerless to penetrate beyond her. That is, everything around you, including yourselves, is a part of nature. And when Darwin wrote on natural selection, adaptation, and evolution, they were not ideas which were easy to accept, especially for people who are steeped in Christian theology. But this is what Darwin said. He says there is grandeur in this view of life, with its several powers having been originally breathed by the Creator into a few forms or into one. And that while this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. This is a sentence which one must read and reread to understand what all he meant when he wrote this sentence. But it is a beautiful sentence because nature continues the process of evolution. But Ronowski, in his book titled The Ascent of Man, also rationalizes the title of his book. He quotes Wordsworth, who said, "For nature to me was all in all." And then Ronowski says this: "Among the multitude of animals which scamper, fly, burrow, and swim around us, man is the only one who is not locked into his environment. His imagination, his reason, his emotional subtlety and toughness make it possible for him not to accept the environment but to change it." And then he says, "And that series of inventions by which man from age to age has remade his environment is a different kind of evolution, not biological but cultural evolution. I call that brilliant sequence of cultural peaks the ascent of man." What are these cultural peaks? These cultural peaks are not what we think as culture. These cultural peaks, each one of them is a technological advance. It's an advance in science and technology at every stage of human cultural evolution. Right from the invention of tools to fire and so on, to the domestication of horses, all the way to the invention of gunpowder, dyestuffs, painting and things like this began only because all of this was possible. Our understanding of the principles of geometry and so on were hugely important. So when Ronowski talks about cultural evolution, he's actually talking about the evolution of the brain, the evolution of our ability to think and our ability to invent things. There are three pillars on which modern biology rests today. Two of them were erected in the 19th century: Darwin's ideas on evolution, variation and selection, and Mendel's ideas of genetics, which is the transmission of characteristics from generation to generation. We all understand genetics. But how is Mendelian genetics linked to Darwinian evolution? After all, children cannot be identical to either of their parents. There's variation. Sometimes there's definite variation between siblings. Understanding variation requires an understanding of mutations and understanding of information transfer in biology. How is that done? That is done through chemistry. So the third foundational pillar of biology is chemistry, erected in the 20th century, when Oswald Avery discovered that DNA is the genetic material. It was the pneumococcal transforming principle in his very first paper. About a decade later, Watson and Crick gave us the double helical structure of DNA, and the double helix is a chemical structure. The ATGC base pairs held by hydrogen bonds are chemical entities. It's the chemistry of DNA which really links Mendelian genetics and Darwinian natural selection. There are two dominant technologies today: information technology and biotechnology. Both of these technologies have their roots in that DNA is the genetic material. This is the starting point of biotechnology, modern biotechnology. Old biotechnology was mostly fermentation. Claude Shannon's work on information theory is the first shot in the information technology revolution. His book, The Mathematical Theory of Communication, is the Bible for everyone working in information science. And interestingly, Shannon's PhD thesis at MIT in 1940 was entitled An Algebra for Theoretical Genetics. And today, if you look at what information science can do, the largest volume of information that is to be handled today is genomic information. Because after the Human Genome Project was completed in 2000, we are now determining genomes by the thousands of human genomes, other organism genomes, and such trees are built. But what can be learned from these trees? From these trees, we learn that humans and chimpanzees are not very far apart. Neither are rats and mice, neither are pigs, cattle, sheep, horses, and dogs. All of us are more or less on the same branch of the tree. What genomic analysis and what I would call comparative genomics teaches us is that there is great unity in biology. Most of the things that you read about in the newspapers and most of the things that you read about every day is about disunity, in thinking that everybody is different from everybody else. Biology does not teach you this. Towards the end of my presentation, I just want to give you one example because I've used the word evolution. One of the most distressing things today in the world is the way in which the word culture is used. Very often the word culture is used in a context by the most uncultured people. Cultural evolutionary theory asks the question, how does culture evolve and does it matter? How does it matter? How does it happen? Cultural transmission is more complex than genetic transmission. It might occur on short time scales, even within a single generation. This is the danger in cultural evolution. But since we are scientists, we can ask a reverse question. Can cultural evolution affect human evolution? That is, can it influence genetic traits by applying Darwinian selection? I want to show you an interesting example. For much of human history, there was little reason to digest milk after weaning. No mammal really drinks mother's milk after it has been weaned, doesn't need milk anymore. It's only right at the beginning after birth. Why is this? This is because adults did not produce the enzyme that digests lactose. The chemical structure is on the slide. The enzyme breaks it down into glucose and galactose. The gene for lactose is shut off during development. Now, if the gene is shut off, there's no enzyme. And if there's no enzyme, you can't digest lactose, so you'll be lactose intolerant. Now, of course, when human beings invented dairy farming, and this might have been 20, 30,000 years ago, human populations had become stationary, they were having livestock, and they began to consume dairy products for nutrition. This happened largely in the northern hemisphere. And when this happened, it turned out that a selection pressure was applied. Those people who could not digest lactose now had severe diarrhea, dehydrated, and probably died. There would be lots of deaths. But slowly over a period of time, human populations get selected out for a trait. And that trait is called lactase persistence. That is, the lactase gene continues to express the protein even as you grow into adulthood. Now, today you can, with modern genomic techniques, go back and ask, is lactase persistence present in human populations? If you go to Scandinavia and Northern Europe, most people are lactase persistent. That is, they can digest milk products very well. If you come to Southern Africa, you will find most people cannot digest milk. If you come to India, you will find most people in North India have lactase persistence. But most people in South India are lactose intolerant. However, because of cultural practices, we have learned to drink milk. We have learned to live with the discomforts that are there. But today, nobody would get dehydrated if they were severely lactose intolerant. So this is an example where an invention, that is the invention of dairy farming, which is an agricultural invention, which is an invention in food chemistry, That now has caused a slow evolution of a human genetic trait. So in long periods of time, and the timescales on which one might think about this grandly are what might happen after 50,000 years, what might happen after 100,000 years of such pressures. So towards the very end of my presentation, I want to show you this. Another book, which if you haven't read it, you should read it. This is written by an Israeli historian, and this book is about human beings, Homo sapiens, and the title of the book is just Sapiens. He talks about history and biology. Yuval Harari, the author of this book, appears to have a better understanding of biology than most people. And he says that the immense diversity of imagined realities that sapiens invented and the resulting diversity of behavior patterns are the main components of what we call cultures. Once cultures appeared, they never cease to change and develop, and these unstoppable alterations are what we call history. When did biology divert from history? If human beings were also hunter-gatherers like all animals, you hunt for your food and then don't do anything else after that. You hunt again the next day for food. You'll be in small groups and you will move from place to place. The cognitive revolution is accordingly the point when history declared its independence from biology. That is the evolution of the brain. And once the brain had evolved, two things had happened. The brain has evolved, agriculture has evolved. Once agriculture has evolved, people became stationary. They have to protect their fields. They have to tend to them. Societies need to form. And once societies need to form, it is important to keep these societies together. And the only way that you can do this is to have historical narratives, which means you should have ideas, you must have images, and you must have fantasies. This is mythology. Now he says this well. He says to understand the rise of Christianity or the French Revolution, it is not enough to comprehend the interaction of genes, hormones, and organisms. It is necessary to take into account the interaction of ideas, images, and fantasies as well. So it is something that everybody should think about. There's a transition from biology to what we are today. And we might ask some hard questions. We might ask, how did life evolve on Earth? How did the chemistry of life evolve? What is the first cell? How did the first cell become a more complex organism? These are the questions on which people work. Those who are interested in evolutionary questions, they ask these questions and work on them today. In my very last technical slide, I end with a question on the coronavirus. You might ask the question, where did the coronavirus come from? Is it natural evolution or is it a laboratory creation? Those of you who read will find that there is a controversy going on in the literature now. Many people think it's the result of natural evolution. Several other people think it's a laboratory creation. Even if not deliberate, it is still a laboratory creation because a lot of gain-of-function research has been done on viruses, where you take viruses and make them now infect human cells. These are to do research for studying vaccines. If such a construct has escaped, then of course there's a danger. And maybe that danger has happened. So this discussion is ongoing. I will not get into this because this is a controversial area. But look at the coronavirus. What does it strike you? If you are interested in symmetry and you're interested in beauty, despite the terrible consequences that happen after infection, as an object, the coronavirus is a remarkably symmetric object. It is, to those who look at only molecules, it is a beautiful architecture. Many, many years ago, in the 18th century, the English poet William Blake actually looked at nature. This is what is important. You must look at nature. He looked at the lamb and he looked at the tiger. Now today, if as a biologist you look at the lamb and the tiger, what do you see? You will immediately see bilateral symmetry. You will see bilateral symmetry even in yourself, the left side and the right side. with an axis going right through the middle of the center. They're beautiful objects. They're still symmetrical objects. And he then asks this question in a poem called The Tiger. He asks, what immortal hand or eye could frame thy fearful symmetry? Then he says, did he smile his work to see? Did he who made the lamb make thee? So we might ask the same question about the coronavirus. Is it made by nature or was it made by man? Today, man is capable of engineering many biological organisms, and therefore one has to really ask this question because the technology really exists. But if you ask such questions, then of course you're being suspicious. And I have myself been accused of being suspicious, but for the last 1.5 years, much of what I've done is to read and to sit in front at my desk in front of a computer and read. And I will end with these two rather wonderful quotations from an author of spy novels, John Le Carre, which I like very much. He says, survival is an infinite capacity for suspicion. And even more importantly, he says, a desk is a dangerous place from which to watch the world. So today, if you look at these controversies in the literature, sometimes you may come up with ideas which are in fact probably considered as dangerous ideas. Lastly, in concluding this lecture, before I conclude, I must apologize to you for the confusion that was there in showing the slides and the fact that the slides did not appear on full screen. This is actually the first time that I've ever experienced this problem, and I don't know its origins. I want to conclude by thanking the two institutions where I have spent much of my professional career and later. The Indian Institute of Science on the top left, where I spent over 42 years, and the National Center for Biological Sciences on the bottom right, which has provided me a home in my retirement years. Thank you very much, and thank you once again for listening to me.
Thank you so much, sir. It was really wonderful and so, I think, very much interesting. We were all glued to the screen. We didn't move left and right. So that is the beauty of, having people like esteemed scientists like Professor P. Balram around. So thanks a lot. Thank you so much for being with us today. So I now request our Honorable Vice Chancellor, Professor Raghavendra Prasad Tiwari ji, to kindly deliver the presidential address.
Professor P. Balram, Professor K. Sambara, Vice Chancellor, Mizoram University, Professor G.D. Sharma, Vice Chancellor, Science and Technology University, Meghalaya, Professor Kori, Vice Chancellor, Central University of Andhra Pradesh, Professor S.K. Mehta, Dr. Raghu Bhaiya, my own colleagues from Central University of Punjab. We have heard very scintillating and inspiring more than 1 hour, 60 minutes talk by Professor P. Balram. I was really mesmerized. For me, this is the first occasion to listen to him, though we are working in a committee. And there also, I had the glimpse of Professor Balram, and that, in fact, inspired me when I interacted with him in the committee. Then that inspired me that I must request Professor Balram to interact with the scientists and teachers of Central University of Punjab, and in the process, we invited several people from across India. We sent invitation to everybody, and I'm sure that after listening to him for nearly 1 hour, several new concepts have emerged about the focal theme of his lecture, that is the chemistry and biology in the age of coronavirus. He started right from the beginning And also, he beautifully expressed how a person who in fact initiated this very branch of science was his contribution was unrecognized, and slowly how what is the history of virus, combining it with biology, with chemistry. When he said that chemistry is everywhere, that reminded me of a stanza from Ramcharitmanas, that is Chit Jal Pavak Gagan Samira Pancharachitaya Adham Sarira. Human body, Homo sapiens itself are made up of the chemistry with 5 elements. And when we see the entire cultural or human discourse with the nature, we all know that the biosphere cannot survive without lithosphere, without hydrosphere, without atmosphere. Chemistry is everywhere, and the civilizational discourse that has happened, in fact, it evolved. Of course, it is biological in nature. It is talking about evolution of organism. So if we go by content or intent, it may appear to be a biological phenomena. But if we see in detail, it has more to do with the chemistry and how chemistry has shaped the entire civilization discourse with nature, how human civilization is surviving, and how it is negotiating with several ailments that are coming, whether man-made or natural. So and alongside, he has talked about variety of other things like culture, and his and how he defined culture, that was really remarkable. And alongside, several other things about space science as well, and how from beginning to the current of coronavirus, and how there is interfacing of biology and chemistry, and how it can make the life of Homo sapiens much more better, and much more comfortable, and much more negotiable with the vagaries of nature, and also with the vagaries of natural viruses or the artificial viruses. Of course, that is debate, and there is no conclusion as yet whether it is really natural or man-made, and that is what is science, because the debate should never end. And because when we debate, when we discuss, newer dimensions of particular science, aspect of science emerge, and that is how the science progresses. We solve one biological phenomena, one chemical phenomena, an event, and other things emerges. So it is almost from every aspect he has dealt with the topic, and he has thrown several new dimensions into the problem, and how the young scientists, the budding scientists, should approach science, and how they should take it to higher pedestal, and how from poets to writers, the best writers. Basically, I am geologist. My specialty is paleontology, and of course, I have read the book that you have referred to, The Sapiens. So this. This shows not how thorough and well-read he. Normally, when we talk about scientists, we say that he talk only about science, but he has touched the human aspects also, and culture is very much human aspect, and also that nothing that exists on this universe is free from chemistry. So this is big lesson, and he has also talked about how. The Nobel laureates, how they have done research, how they have really got Nobel Prize, they awarded Nobel Prize with interfacing of biology with chemistry, and even in biology, biochemistry, biotechnology, and almost all branches of biology spoke about, and also the chemistry, how these two beautiful sciences can join hand together to solve several issues. And not only that the human civilization is currently confronted with, but also that are likely to emerge. So we are really thankful to you, sir, for delivering very nice, very inspiring speech. And I'm sure that the listeners were like me, all the listeners were mesmerized, and they have really, some new ideas have emerged. And and particularly the young budding ones, they will pick up some of those ideas and they will pursue that as their future career. And also at the end, the last slide where he worked all through acknowledging the alma mater, the parent organizations. These two organizations, the first one has really made you, Professor P. Balram, that we know today, the scientific community know today, and also where post-retirement shelter. So the human touch that you have given to this, that was really remarkable. So I'm again thankful to you. And when we are free with coronavirus, I don't know whether we will be really free with coronavirus, but when it subsides, then I will request you to come to us personally and interact with our, we are very young university, as you know. This university was started in 2009, and the average age of teachers here is around 40 years. They are young, all young budding scientists, but if you see their publication profile and all, we are really good in the 5-point scale. We are good, and our endeavor is to become excellent. And and we intend to be a research-specific university right from the start, right from the founding vice chancellor of this university. The effort has been to on this research-specific university name to this, and this also syncs very well with the new education policy. So we want to further advance in the area of research, and for this, I most sincerely wish that you come to us, interact with our budding scientists, you see what actually they are doing, what is their potential to do, and what they should be doing next. So thank you, sir, for our accepting our invite and making the day for not only for Central University of Punjab teachers, but also teachers from elsewhere, really a memorable day. Thank you, sir. Jai Hind, Jai Bharat.
Thank you so much, sir. Now I request our Dean in Charge Academics, Professor Ramakrishna Musrika, to kindly propose formal vote of thanks.
Good afternoon, everyone. So we would like to thank Professor Balram for his fascinating account of scientific history of coronavirus. So today we came to know a number of new things about coronavirus, from where it originated, because whatever we have read was quite different from what account he gave, going back to many years before than what we generally read in the literature. So that was really useful for all of us. And then we also were told about chemistry. Like, we all know how important it is, chemistry overall. So that was something we all work with. Another aspect is how these days science is done in a transdisciplinary manner, kind of thing you highlighted very well. So it is not only biology, chemistry, evolution, but also you try to link that with culture and other aspects. I think this is a holistic way of doing science. I think that is how we should all go these days, because 20-30 years it was different. We sit on one molecule and work on that, but now it is quite different. So that is something highlighted from his talk. So overall, I think it was very informative kind of talk, which was, we are all fortunate to hear, and your talk. So number of us have heard your name a lot, but we have never heard you talking. So this is the first time we heard. So thank you very much. And I would also like to thank Professor Tiwari. He is the one who has been pushing for these kind of seminars with pioneers like you. So it is really good to hear somebody from biochemistry background, and Dean Research for organizing all the seminars, and all my colleagues who came here forward to listen to this. And I can see that the Google Meet is overflowing. So we try to create a parallel site on YouTube so that more people can benefit from your lecture. Thank you very much.
Thank you, Professor Ramakrishna. So we all stand for national anthem.
पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल गंगा विंध्य हिमाचल यमुना गंगा चल जगति तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे गाहे तब जय गाथा जय गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
So thank you, sir.
Thank you,
once again.
Thank you. Bye. Bye.
Yes.